working in the lab is not horrible. It's the best thing. It's the best thing, especially the microbiology lab class. That's what everybody says that at the end of our student questionnaire. Well, for this course, you know. Yeah. So tell me at the end if you hated it or not. Right. Okay, well, we have a couple of minutes before everybody arrives, I guess. So I'll just take some time to explain to you why I'm so tired today. I'm so tired this morning because my dog woke me up at 1:30 in the morning, kind of downstairs, barking a little bit, whining at the door, and I kind of thought, oh man, because he doesn't usually do this, right? So me and my wife were lying there and thinking, what was it? What's going on? Maybe there's somebody outside there coming to rob us or something, you know. So I kind of sneaked downstairs, didn't put any lights on in the house so that they didn't like see me, and then like put the light on in the garden and looked, checked to see if there's anybody there. Nobody there. Okay, I let the dog out. He went out, stayed there for ages, and I was like, oh, come on, come on. And then he came back and brought him back in again, and went back to bed. You know, 15 minutes later, the same kind of thing, down at the door again, whining, barking. Oh no, come on, what is it this time? So I let him out the door again. You know, checked outside to see if there was anybody there. Let him out. He was out there. He stayed for ages, and I had to go out in the garden and get him. He was just sitting there doing nothing, standing on the steps. And then I kind of like got scared. Oh man, this is some kind of ghost or something. He could detect paranormal activities in our house, this guy. And I was like, so in the end, got him back in and put him in his little basket and went to sleep. Like, oh my God, it's just like the film. There was a dog in that as well. So come down in this morning. You know, my alarm goes off 6:15. Get a shower, come down. Then I realized, what did I find downstairs? It was not any supernatural activity. I just saw a big pile of dog vomit in the kitchen. So that's why he was so upset. He was really, really sick, the poor guy. So that's not the best thing for me on the first day of term when you have a class at 8 a.m., I must say. So if I'm looking rather tired and start falling asleep halfway through my own lecture, then that's the reason why. Okay. Right. Well, that was kind of random introduction. We're not really going to be talking about dogs all the way through the rest of this course, but we might be talking about foodborne infections. So welcome everybody to the L2 ABT and specifically to microbiology. And as we said earlier this morning, there are some people from all over the place, which is nice for us. So please help people fit in and enjoy living in Nantes. Now for the microbiology class, this is a 40-hour course and we have 20 hours of lectures delivered by me and Madame Moratou. 8 hours of, well, TD is what I would call maybe problem-based learning classes, and that's Madame Moratou, and there are 3 4-hour lab sessions, and that will be me working in the lab class. Now, as well as that, there are some kind of distance learning activities on Madoc. So there are some short videos to look at before the lab classes, together with a short, very easy quiz, just to test if people have been looking at the videos and watching them. And the idea is that people there are demonstrating the same techniques that you'll be using in the lab class. Now, of course, I'll be showing you in the lab, but you know, it helps if you've already seen it once, I think. The other thing is I record the lectures, so afterwards you'll be able to listen to any parts of the lectures again if you feel that it's not clear, or if you want to like check that you heard correctly what I was saying. So those should be available. And the other thing that I do with ABT is microbe of the week. So basically once or twice a week, I will send out a question in the evening by email. So we'll start with one this evening at 7 p.m. And the idea is that you all go find the answer to the question and write in your own words the answer and email it back to me. Okay, so of course it's quite easy to find some kind of answer, copy the text and send it, paste it in the mail. But that's not the point. The point is for you to like try and find some scientific information and then write about it in English. Okay. Now there's no credits for this in the module. It's not evaluated, but there are prizes. Okay. So it's basically 1 point per correct answer. Sometimes there's a bonus question and there's like 3 points for the first person to answer correctly. Okay. So it's kind of a race. So that's why at 7 p.m. I send the question out. You all be ready to get the answer and send it back to me. So there's a kind of scoreboard, and at the end of the semester, there's prizes for the winner. Sometimes second and third place as well. Depends, depends how close it is. All right. I mean, if there's 2 or 3 people like way ahead, then you get some small prizes. They're real prizes, right, Ilana? Yeah. Okay. Now, for the evaluation, it's all 60% on the exam at the end of, well, in January, and 40% on, well, other types of evaluation. So 20% on the lab report, 20% on the written test, which is at the end of the semester. Now, if you like microbiology, so we're changing the undergraduate program next year, and what is currently the plan is to have half a module in virology and mycology, so that mostly you're doing bacteriology this year and everyone gets a chance to do virology and mycology next year. And then there'll be an option in semester 6 about microbes and health. As for other reading, then I'm a big fan of textbooks. Pretty much everything we cover in the lectures this semester is explained very, very clearly in any standard textbook of microbiology. So in the library, we have Prescott's microbiology and Perry's microbiology. So once again, if there's something you don't really understand in the lectures and it's still not clear if you listen to what I'm saying again, try and look in the textbook. It's someone else writing about it, explaining things in a really clear way, well illustrated. Now, other things you might want to check out. Oh, yeah. So the ASM blog here, this is quite a nice thing. American Society of Microbiology, an old retired microbiologist giving like news about recent, well, information in microbiology. This is an online textbook. This Week in Microbiology, great podcast series co-hosted by Vincent Racaniello. I don't really listen to it much myself. I'm a bit of a This Week in Virology person, to be honest. But it's probably well worth checking out. Now, a few years ago, okay, so this is basically the program of what we're going to be covering over the course of the lectures. So this afternoon, we'll be going through the structure and functions of the bacterial cell. What is it? It's Thursday, right? We see each other again. Or Friday. Anyway, third lecture this week, bacterial identification and taxonomy. Then we'll be looking at more bacterial physiology, growth, the impact of environmental factors, and then bacterial nutrition and energy metabolism. One lecture on antibiotics, then a bit about bacterial genetics, interactions between microbes and their hosts in a positive and a negative way. We'll have a couple of lectures on viruses and something about applications and biotechnology. So that's the program. Now, we don't cover the whole of microbiology in this course because microbiology is basically anything that's microscopic that you can't see with your naked eye. And this includes, of course, a lot of microscopic eukaryotes. Now, we're not going to cover these at all. Okay, so most of the time we're going to be looking at bacteria, and we'll have a little bit about viruses. And among bacteria, most of the time we're going to be concentrating on eubacteria, which is true bacteria, which are very different from archaebacteria, archaea. Now we might mention archaebacteria from time to time, but we don't really cover them very much. So most of the time during the lectures, when I say bacteria, what I really mean are these guys. Okay? And these are the vast majority of bacteria that you will encounter in health and disease, in our microbiome rather, and bacteria that you can isolate from the soil, stuff like this. They are almost always going to be eubacteria. Archaebacteria, many of them are limited to extreme environments like hot springs, very acidic environments, but there are a few that are strictly anaerobic gut bacteria. So methanogens, for example, are archaebacteria. But most of the time, we're not covering these. So bacteria, eubacteria, what is so great about them? Why is it so important to study bacteria? Well, one of the reasons is because they are everywhere. They're in every environment on Earth, all over the place, in us and on our skin. And even in places where you probably wouldn't expect anything to be living at all, like in a hot spring at 95 degrees Celsius, pH 2, there are some bacteria that can live in those conditions. There are bacteria living underneath frozen lakes in Antarctica. Now, you kind of think, how on earth can they possibly live there? Well, they can do this because they can extract energy from pretty much any kind of chemical source and also photosynthesis. So they can live on practically anything. And they can do this because they can harness a lot of interesting biochemical reactions that we can't do. Eukaryotes, vertebrates, vascular plants, we can't do this stuff. So there's a whole range of metabolism that's present in bacteria that doesn't exist in eukaryotes. And some of these are useful for industrial applications. So that's one reason why it's important to study bacteria. Of course, there are several bacterial pathogens that are still very, very important and have a huge impact on health. So Mycobacterium tuberculosis, I haven't looked at the numbers recently, but it's probably the case that it's tens of millions of people worldwide infected by this bacterium, and it's probably responsible for maybe 1 to 2 million deaths per year. So this is a significant burden of disease. Same for things like Salmonella, Staphylococcus. There are a lot of very frequent infections caused by bacteria. Now, the other flip side of that is that our bacterial microbiome is a very important part of our, physiology. And if you ignore the interaction between the host and microbiome, then you're not really understanding everything that's going on. Now, of course, bacteria are very important symbiotic organisms, not just in humans, but all over the place. So nitrogen fixation in leguminous plants is entirely dependent on their symbiotic bacteria. And this is extremely important for food production and agriculture. And maybe the last thing here in this introduction is that bacteria are, of course, the simplest independent living organisms. And if you want to understand the fundamental processes of life, like DNA replication, protein synthesis, all of these things were studied first in bacteria because they are the simplest model system that you can study. And because bacteria are the simplest living organisms, if you want to understand something about the origin of life, you have to think about the origin of bacteria. So looking at these simple organisms, thinking what's the actual minimum that a cell needs to survive, gives you some idea about the very early stages of life on the planet. Okay, so that's basically the introduction. Now we're going to go into how to study bacteria. Well, the first thing that you need to be able to do is to observe them somehow. So you need a microscope. And if you have a good phase contrast microscope, you can see bacteria, oh yeah, without actually any staining agent. So you can see, you've got these bacterial cells here. You can have an idea of their size and their shape. Sometimes for a particular bacteria, you can see that some bacteria have inclusion bodies inside them, these very refringent. These are granules of sulfur in these bacteria. Now, most of the time, and you'll see this in the lab class, it's not so easy to actually see bacteria in a microscope without staining them. So coloration is an important part of observing bacteria just on a standard upright microscope. And so there are simple stains like crystal violet or methylene blue, which will just stain every type of bacterium. So you just get a little bit of information, their size, their shape. And here with this one, you can see that the bacterial surface isn't entirely smooth, but there's a kind of like halo around the bacterium and a kind of slightly colored region around the side, around each cell. So this is what's called the bacterial capsule. Sometimes it's visible on light microscopy. Another thing that is also very useful are differential stains, which will color different types of bacteria differently. Now, the most frequently used differential stain is, of course, gram staining, which will color gram-positive bacteria dark purple and gram-negatives are pink. So somehow you can see that there's some difference, and even though they might look quite similar, they're not all the same bacteria. Now, another differential stain which is very useful is acid-fast staining or Zeil-Nielsen staining, which is used to stain mycobacteria. So here, these pink rods, they're mycobacteria, and it's very important for screening for tuberculosis. There is still acid-fast staining on a sputum sample from someone who may be infected. Is the frontline screening technique still. This is like probably more than 100 years old, this technique. And other stains can be used to identify endospores within certain types of bacteria. Okay, so you look at bacteria in the microscope, perform a gram stain, and you can identify different shapes of bacteria. So if you've got pure bacterium from one colony, you'll notice that every single cell has the same shape. So the shape and the gram stain of the bacterium is an indication of its identity. What is this bacterium? So the two shapes that are the most frequent are cookie and bacilli. So spherical cells or elongated cells. And of course, cookie can be isolated, diplocookie, or in bunches or in chains. Now for bacilli, that is rod-shaped bacteria. And they can either be, clearly like, rods, or sometimes you get what are called cucurbacilli. They're shaped a little bit like a rugby ball. It's just slightly oval. But this is still, bacilli. It's not the same thing as a perfectly spherical cell. So these cell shapes are the most frequent that you see, but there are a bunch of other types of bacteria that have a very characteristic shape. So Vibrio are gram-negative bacteria that are shaped like a comma. These bacteria with kind of pointed ends are Fusobacterium. All of these kind of helical gram-negative bacteria are spirochetes. So Borrelia is a very typical spirochete. So shape and gram stain are basically the first things that you're going to look at if you want to identify bacteria. It'll give you some kind of idea of the genus, not the species, maybe the family or the genus of the bacterium. Now, if you want to look a bit more closely at bacterial cells, you need to use microscopy, which is more powerful. So electron microscopy. And this can either be scanning electron microscopy, so you get an idea of the surface of the bacteria, or transmission electron microscopy. Now, in transmission electron microscopy, you can do two things. Firstly, you can take an ultra thin section through the bacterial cell and stain what is inside the cell. And here, for the first time, you get an idea of what the inside of a bacterium looks like. And it's very simple, okay? You've got a kind of cell wall on the outside, which is a little bit opaque here. Then you've got a cytoplasm with lots of ribosomes in it, maybe some inclusion granules, and what's called the nucleoid, where the DNA is concentrated. But you don't have any membranous organelles inside the bacteria. The DNA is directly in contact with the cytosol. So you get a look at the inside of the bacterium. Now, another thing that you can do with electron microscopy is fix the bacterial cell to some kind of inert surface and then try and shade all the structures by, I believe, evaporating heavy metal ions from one side of the grid. So anytime you've got some kind of like physical structure that's on the grid, you'll get an accumulation of heavy metal ions that you can see here. Okay, this black stuff here, this is where some kind of like metals have accumulated next to this structure. And you can take a picture of this. For the first time, you can see that the surface of the bacterium isn't really smooth like here. It's actually quite hairy, right? So we have certainly at least two different types of filaments here. You've got this big, long, thick one, which is a flagellum. Here's another one. And you've got lots of slightly shorter, narrower filaments, which are called pili or fimbriae. So that's kind of a view of the surface of bacteria and on the inside of them. Now, if you want to go a little bit further and understand the biochemical or the molecular structure of these things that we can see on electron microscopy, there are two kind of approaches. So one of them is a classical biochemical approach. Like, I want to know what the external membrane of a bacterium is made out of, a gram-negative bacterium. I'll get my bacteria, I'll dissolve the external membrane, smash it up, and then purify the different components and characterize them biochemically. And that's how we know that the external membrane of the gram-negative bacteria is composed of lipopolysaccharide. And so you know you've got this lipid part here and then a whole bunch of sugars forming the polysaccharide bit of it. So biochemical approach. Another kind of approach is if you want to, for example, understand about, I don't know, what is this made of, this filament? What you can try and do is generate a panel of mutants, find a mutant that doesn't produce these things anymore, clone the gene, sequence it, and then from that you'll be able to perhaps express the protein, analyze its structure, and then you'll get a model of the proteins that go and make up, for example, this these pili. No, well, it's not the example that I have on the slide. The example I have on the slide are these membrane porins, which are also found in the outer membrane of gram-negative bacteria. So they're kind of like barrels made up of beta sheets, and each one of them is made up of and the functional protein is a trimer or homotrimer. And so you can see that, this creates a kind of empty space here, and small molecules can diffuse through these pores in the outer membrane. So bit by bit, if you combine the genetic, structural data and the biochemical data, then you can build up a pretty accurate model of the structures that make up a bacterial cell. So this one, for example, is a model of the base plate of gram-negative flagellum. So you have each protein is colored in a different color, and they are represented to scale with the outer membrane, inner membrane here, and peptidoglycan layer. So this is kind of nice, pretty looking picture, but it's not just kind of how I imagine this thing looks like. This is actually a real representation of the structure that actually exists. And we know it because probably hundreds of people have been working on different aspects of this problem for decades. And that's how we have a really good, accurate structural information about all these elements of the bacterial cell. So from now on, when I'm representing stuff in the bacterial cell, it will be some kind of very, very simple representation, like a little rectangle or an oval or something like that. But try and bear in mind that behind that, there's actually a lot more information that we know about these structures. Okay, so it's time for me to have a very short break and see if you've been listening. So if you flash this code here, it should take you to a page on Madoc where I'll be able to open up this question. If, okay. Oh, it's blocked, it's not doing anything. Okay, should be ready to go now. Okay, that's about it. Right, so Okay, here are the votes. So capsule, yes, that's correct. Pili, no, no way. These things are about 3 to 10 nanometers in diameter. You can only see them by electron microscopy. LPS, no. LPS is a molecule. Lipopolysaccharide is a molecule. You can't see LPS any more than you can see a molecule of glucose in a microscope. Everybody missed this one, the endospore. Yeah, that's big enough to see by light microscopy. It's big enough. Nucleus, no, no bacteria has a nucleus. No bacterium has a nucleus. So correct, wrong, wrong, correct. Okay, so that gives me perhaps the excuse to briefly remind everybody about the main differences between eukaryotic and prokaryotic cells. So in eukaryotes, there are a large number of organelles, Golgi, mitochondria, chloroplast, the endoplasmic reticulum, and they are all bound by lipid membranes. So in prokaryotes, there's really no organization inside the cell, maybe with the exception of inclusion bodies. But these inclusion bodies don't really have membranes around them. Okay, so these organelles kind of communicate with each other through the cytoskeleton in eukaryotic cells. There's not really very much cytoskeletal organization inside bacteria, but there are a couple of structural homologues of tubulin and actin that do play important roles in bacterial growth and division. So they have some kind of like simple precursors to the complex cytoskeleton that you see in eukaryotes. So in bacteria, there's a rigid cell wall outside the plasma membrane composed of peptidoglycan. Now, some eukaryotes have a cell wall, but the chemistry is very different. So they don't have a common origin, these structures. The external membrane on the outside of the cell, there's only one in eukaryotes, the plasma membrane. In bacteria, there can be one or two. So gram-positives just have the plasma membrane. Gram-negatives have the plasma membrane and the external membrane. Pili, these protein structures around the outside of the cell, very many of them in bacteria, not in eukaryotes. Flagella, okay, sometimes in prokaryotes and eukaryotes. But once again, if you look at the structure, there's no homology between them. So these are both, so the flagella in eukaryotes and prokaryotes have the same function, that is to move around in a liquid medium. But they have evolved independently. So that's why they don't have any structural homology. So it's an example of convergent evolution. Okay, the capsule, this kind of layer outside a bacterium, very often in bacteria, don't really have any equivalent in eukaryotes. Spores, that is a very resistant cell that is metabolically inactive. Yeah, fungi and plants produce spores. Some bacteria produce spores, but again, structurally, they're quite different. So it's independent solutions to the same problem of surviving in very difficult conditions. Now, there are a lot of differences in the way genetic information is organized inside eukaryotes and prokaryotes. So eukaryotes are all either diploid, or at least they have a diploid phase in their life cycle. Okay, so lots of eukaryotes that are haploid and diploid, but at some point they can become diploid. All bacteria are only haploid cells. Now in eukaryotes, the chromosomes, there are several linear chromosomes in the nucleus, whereas prokaryotes generally just have one circular chromosome. Now this circular chromosome is very often accompanied by smaller circles of DNA called plasmids inside the bacterial cell. And there really aren't any plasmids in eukaryotes. Okay, nucleus, yes, in eukaryotes, no in prokaryotes, and DNA is condensed in eukaryotes by histone proteins, which are not present in bacteria. Okay, so there are maybe some exceptions. So for example, bacteria that have 2 chromosomes are not that rare. So Vibrio cholerae is an example of a bacterium with 2 chromosomes. And in some types of archaea, DNA is associated with histones. So this is some indication that eukaryotic cells and archaea have some kind of common ancestry. That's one of the indications. Okay, now this is my, now the more I look at it, the more I think this is a really terrible diagram that I drew of a bacterial cell. But what it's supposed to illustrate is, well, you know, the basic structures that you can find in bacteria. Now, every time we've got something that's underlined, that means this is an essential structure that you find in all bacteria, all eubacteria. Plasma membrane, peptidoglycan cell wall, and a nucleoid. All the rest are facultative structures. So the external membrane, only in gram-negative bacteria. The capsule, which is a layer outside, some bacteria have it. Flagella, once again, facultative, Fimbriae pili. I've put 2 different colors here to show that there are different types, but some bacteria have none. And inclusion bodies might be present, might be absent. So what I'm going to do from now on is go through these different structures, and basically I think we're going to finish this afternoon looking first at the external structures and then the internal structures. And I'm going to start with the cell wall. Now, bacterial cell walls in eubacteria all contain peptidoglycan, and the peptidoglycan layer is what gives rigidity to the bacterial cell, because bacterial plasma membranes are very fluid. Okay, now, so if you just have a bacterium without a cell wall, it can survive if it's in a liquid environment that is isotonic. Okay, so the osmotic pressure outside the cell and inside the cell is the same. So that way the cell will keep its shape. But if you don't have a cell wall and you're in a hypertonic environment, which is most of the time, maybe not inside the sea, but then water is going to diffuse into the cytoplasm and it will explode the cell. So in order to avoid this happening, all bacteria have got this rigid peptidoglycan layer outside the plasma membrane that allows the cell to resist osmotic stress. Now, the basic structure of peptidoglycan is conserved amongst all bacteria, and that makes it a good target for antibacterial molecules that are produced by eukaryotes or other bacteria. So, for example, lysozyme, all vertebrates produce lysozyme. It's an enzyme which will break down peptidoglycan. So it's part of our innate immune resistance mechanism to avoid getting colonized by bacteria. Beta-lactam antibiotics such as penicillin are produced by fungi and other beta-lactam. Beta-lactams all produced by fungi. So, okay, fungi don't have a peptidoglycan cell wall, so they aren't susceptible to antibiotics that inhibit cell wall synthesis, peptidoglycan synthesis. And the other thing that is important, well, the most important thing to remember is that even though gram-positives and gram-negative bacteria both have a peptidoglycan layer, the way it's organized is very, very different in gram-positives and gram-negatives. Now, to understand the main difference in the organization of the cell wall between gram-positives and gram-negatives, I'm just going to take a little time to explain how gram staining works. So the first step, as you all do in the lab classes, which everyone is going to love, right? The first step is to fix the bacteria onto a glass slide and then stain all the cells with a non-specific stain, which is crystal violet. So in the first step, all of the bacteria will be stained purple. Now, crystal violet is a basic stain, so it's got positive charge, and it will bind to anything with a negative charge. So what's it binding to in here, inside the cytosol? No, gram minus and gram positive both get stained. So what kind of molecule inside cytosol has got a negative charge? Molecule. Got excess electrons associated with it to stay negative, yeah. Okay, something with a positive charge is going to be basic, with a negative charge is going to be acidic, right? So something acid inside cells. What is this supposed to be? What is that supposed to be? No. Maybe the other side. What is this red thing here? That's the protein. This is RNA, right? RNA means what? Acid, right? Ribonucleic acid, right? All nucleic acids have a negative charge, acid, because of the phosphate in the backbone. So all of this basic stain, crystal violet, will have some kind of just electrostatic interactions with the phosphates on here. Okay, negative charge, positive charge, it will bind. So it's very non-specific. Any kind of cell gets stained here. So that's the first step. Second step is to add in some iodine, and this will form big complexes with the crystal violet and all the nucleic acids, and that won't really get washed out easily. Won't get washed out with water, for example. Now the next step, the third step, is to wash the cells on the slide with alcohol. And what happens here is that some of these bacteria lose the stain, and some of them will keep it. And these ones that lose the stain in the alcohol wash are gram-negative stain cells, because they lose the gram stain. Whereas the ones that retain the gram stain in an alcohol wash are gram-positive. Now at this point, these cells become invisible. They don't have any crystal violet inside them. Okay, if you notice, I'm looking around in a rather perplexed way, because what am I looking for? There's nothing here to wipe the border anymore. Anyway, so in order to see these cells, the last step is to add a counter stain, which is another coloring agent with a different color. Okay, so it's often fuchsin, which will stain cells light pink. So the main thing to remember about this is that the big difference between gram-positive and gram-negative cell walls is that gram-negative cell walls are sensitive to alcohol. gram-positive cell walls are resistant to alcohol. Okay, now there are some exceptions. Most bacteria, you can perform gram staining and you can identify them as gram-negative or gram-positive. So there are some exceptions. So mycobacteria have a very distinct cell wall structure. They've got an outer membrane, but it's very, very different from what you have in gram-negative bacteria. So they don't really work in the gram classification. And there are also some bacteria which do not have any peptidoglycan at all. And these are the mycoplasmas. Now, that might sound like it shouldn't exist because I started off this whole segment by saying that the peptidoglycan cell wall is absolutely essential. But these bacteria can somehow survive without it. And they can survive because they have a very particular ecological niche where they live. They are all parasitic bacteria that live on the outside of eukaryotic cells. So for example, our cells, we don't have a cell wall, right? But our plasma membranes are slightly more rigid than a bacterial cell plasma membrane. Why is that? What do we have in a eukaryotic cell membrane that solidifies the... No, I'm talking about what molecule, specific molecule that is in part of our membrane, plasma membrane, but only eukaryotes can make this, bacteria can't make it. Nope. No, okay, no, good try, but bacterial membranes are also made out of phospholipids. So in our membranes, we've got phospholipids and something else which adds rigidity. Nobody? Do you want a clue? Yes, cholesterol. Okay, so that's why our plasma membrane can actually resist a bit of osmotic stress, the cholesterol. So these guys, mycoplasmas, they live on the outside of eukaryotic cells and they can extract the cholesterol and integrate it into their own membrane. So they get their plasma membranes have got the same kind of structural rigidity as ours, but they can't synthesize it, so they get it from their host cell. So that's an exception, but it's quite rare. Okay, so most bacteria living around freely, they can't do that. So for them, the peptidoglycan layer is absolutely essential. Okay, so let's get back to this question about alcohol sensitive and alcohol resistant. So if you look at the cell wall of a gram-positive bacterium, which is over here somewhere. Oh, where's my arrow? Yeah, gram-positive here. You can see you've got the plasma membrane here, and then you've got a very thick layer of peptidoglycan. And this will resist alcohol. Whereas if you look at a gram-negative cell wall, you've got the plasma membrane, a very, very thin layer of peptidoglycan, and an outer membrane here. So if you look at the schematics here, in a gram-negative cell wall, you've got outer membrane, inner membrane, and a very thin layer of peptidoglycan. This is sensitive to alcohol and to detergents. So during the alcohol wash, it just gets removed. And the peptidoglycan layer here is very thin, so it doesn't resist very much either, and all the stain gets washed out. In gram-positive cells, you've got a very thick layer of peptidoglycan, and this is what resists the alcohol. Okay, it's not soluble in alcohol. So that's really the big difference in structure between gram-positive and gram-negative cell walls. Never ever forget this. Gram-negative outer membrane, thin peptidoglycan layer, gram-positive, very thick peptidoglycan layer. Okay, peptidoglycan. Now I've been talking about using this word, and it's very important to understand the structure of this molecule and why it's so useful for bacteria. Now, peptidoglycan, the clue is in the name. You've got a glycan part and a peptide part. Now, the glycan part is composed of two modified sugars, which are N-acetylglucosamine, which I often call NAG, N-A-G, which is connected to N-acetylmuramic acid by a beta 1 to 4 linkage. So I don't know if you remember sugar structure, okay? Going to be something like this, and you've got carbons 1, 2, 3, 4, 5, and 6 is up here. So beta 1 to 4 is going to be, look, this is number 4, so it's going to be something like this. And beta is to do with whether this one's going up or down, something like that, okay? It's biochemistry. But basically, it's that kind of idea. These two are derivatives of glucose. So N-acetylglucosamine, which is going to be here. So glucosamine is glucose with an amine group at position 2. And N-acetylglucosamine has got an acetyl group here. So that's on position 2. N-acetylmuramic acid is the same thing, except that it's got a lactate group stuck on here on carbon 3. So lactate is going to be something like CH3, CH3C, and then O, and then O my. So something like that, okay? It's kind of stuck on here on carbon 3. And that's why it's called muramic acid, because it's got this carboxylate group here. I think that's wrong, isn't it? It should have 3 carbons. That's more like it, yeah, like that. So that's why you've got an acid group here, carboxylate on the N-acetylmuramic acid. So the glycan part is just like a big linear chain of these two modified glucose molecules, and it will give you big, long linear chain. Okay, NAM, NAG, NAM, NAG, NAM, NAG, NAM, NAG, big, long line of polysaccharide. So these form filaments. Now, if you want to get a structure that is solid, that's not enough, you know, because you would just be like if you had a load of threads like laid alongside each other. But that's not going to resist osmotic pressure because it would just separate the threads. So to get a structure that is actually resistant, you need to join these threads together, and that's the function of the peptide part. So peptidoglycan, the peptide part, are 4 amino acids which are stuck onto the N-acetylmuramic acid. How are they stuck on there? Well, you've got this carboxylate group, and this can form a peptide bond with the N-terminal amine group of this first amino acid. Okay, so one of the things you need to remember is the tetrapeptide, is it attached to the NAG or the NAM? It's the NAM, because you need the acid in order to make this peptide bond here. Okay, so these, you've got 4 amino acids stuck to each N-acetylmuramic acid, and the third one is cross-linked to the 4th amino acid on another chain of NAM-NAG. So this introduces a lot of cross-links in the structure. So that means instead of just having a load of filaments like that, you've got some kind of net, okay, with the mesh of the net, which is quite a small structure. So that means these filaments, they can't be pulled apart, okay? And also, this is just a 2D representation, but you have to imagine you've also got filaments lying on top of the screen here and behind it, and the crosslinks can go in 3 dimensions. So the whole thing becomes quite rigid and very resistant. And the mesh size here is quite small. A protein can't really diffuse across peptidoglycan. Small molecules like glucose, disaccharide, and amino acid, they can just diffuse through. But proteins and things of that size, they can't really get across. Okay, so that's very, very solid structure. Now, there are some differences in the biochemistry of peptidoglycan between gram-positives and gram-negatives. So, for example, in a gram-negative like Escherichia coli, the third amino acid here is diaminopimelic acid, called DAP, and the cross-link to the fourth amino acid, which is D-alanine, is direct. Whereas in many gram-positives, like Staphylococcus aureus, the third amino acid of the tetrapeptide is lysine, and it's connected to the D-alanine by 5 glycines. So that kind of makes sense, right? Because in gram-positives, the peptidoglycan layer is very thick. So because of this glycine bridge, that means that filaments can be connected from across a greater distance. Whereas in gram-negatives, the two NAG chains that are connected by this peptide bridge are going to be very, very close together. But that's okay because the whole peptidoglycan layer is very thin. Okay, so some differences in the biochemistry of peptidoglycan between gram-positives and gram-negatives. Now, I want you to take a moment to think about the particular amino acids that make up or that are incorporated into peptidoglycan. Oh, hang on, let me just reinitialize and start. Okay, should be okay. So what is unusual about these amino acids? Yeah, there's a lot of biochemistry in microbiology. Okay, that will do, I think. Okay, so here are the answers. Answer number 4. No, that's just a trick question. It doesn't really mean anything. So that's not true. Answer number 3. Yeah, that's correct. I mean, DAP. Has anybody heard of this thing before today? No, it's not one of the 20 amino acids that are incorporated into proteins. It's something different. And 1 and 2. Well, one of these is correct, and the other one is wrong, because they're like inverses of each other. And the correct answer is 1. Okay, in proteins you only have L amino acids. Okay, so it's D glucose, but it's L amino acids in cellular metabolism. So these two things, the fact that you've got D amino acids and the fact that you have an unusual amino acid in the chain, the DAP, are kind of suggesting that the peptide part of peptidoglycan is not synthesized on the ribosome. It's not the same thing as a protein. So there must be a different way to synthesize this peptide. Now, for the next section here, I'm going to try and explain the different steps in peptidoglycan synthesis. And as I'm sure you'll appreciate, it's going to be a little bit complicated. So the first thing the bacteria do is synthesize the repeating subunit in the cytosol. So the repeating subunit is the NAM-NAG tetrapeptide, but in the cytosol they produce a pentapeptide. And we'll see why that's important later on. Then, once the subunit's been synthesized, it's flipped over into the periplasm. The periplasm is the space between the plasma membrane and the peptidoglycan layer. And it's flipped over in the plasma membrane by this lipid transporter, which is called bactoprenol. So that's good. The subunit NAM-NAG pentapeptide is in the right place. Then, firstly, transglycosidases will insert it into the peptidoglycan polymer. And the last step is the formation of these peptide crosslinks by transpeptidases. Now, when bacteria are growing, that means the cell size is getting bigger, and then at the end, the cell will divide in two. But in order for this bacterium to grow, it's got to be able to insert these subunits into a polymer that already exists. And in order for that to happen, growing bacteria have got to cut the polymer up at the same time and then insert new subunits. Okay? So there's an equilibrium between synthesis by these transglycosidases, transpeptidases, and destruction of the peptidoglycan by enzymes which are called autolysins. And that's the reason why antibiotics that inhibit peptidoglycan synthesis are bactericidal. So penicillins, cephalosporins, they inhibit the transpeptidase step. Vancomycin also inhibit the transpeptidase step, but by a different mechanism. These are bactericidal. Now you might wonder, well, why is that? Because they're just like inhibiting the last step. But if these crosslinks aren't made, then at the same time you've got these autolysins that are breaking everything up, then that's why the cell will explode. Okay, because you prevent the synthesis from being completed, and at the same time bacteria are breaking down their membrane, their peptidoglycan. So they are bactericidal, and they will work better on bacteria that are growing, actually. And you'll be able to see that in the lab class as well, hopefully. Okay, so those are the different steps here. And you have to try and wonder, well, this is something I do. Whenever I see something in biology that's complicated, and there's a lot of complicated things in biology, you have to try and think, why is it like this? Why? I mean, wouldn't it just be simpler if the bacteria just exported the NAM, the NAG, and the amino acids outside and then like put the whole thing together where it's supposed to be? Well, the problem is any kind of biochemical synthesis requires you to put energy in to make these bonds. And on the outside of the cell, you don't have any ATP or any source of metabolic energy. So it's really a complicated problem. How do I make a complex molecular structure outside the cell? Because outside the cell, you don't have any metabolic energy. And The reason the synthesis pathway is so complex is because bacteria have to use some clever tricks to solve this problem. Okay, so let's have a look how it works. So the first step is to synthesize this kind of molecule, N-acetylmuramic acid, um, well, bound to or combined with uridine diphosphate, UDP. And as you remember, N-acetylmuramic acid has got this lactate group attached onto the carbon 3, and the carboxylate group allows the attachment of these 5 amino acids and a pentapeptide. So this is the N-terminal part, the L-alanine, and it's got an amine link to this carboxylate group here. And then you've got like N-terminal, C-terminal, N-terminal. These are just peptide bonds all the way down to the D-alanine here, and this one's got a free carboxylate group. Now the first thing that happens is that this NAM pentapeptide is going to be transferred onto bactoprenol, which when it's facing the cytosol is present as a monophosphorylated bactoprenol. Now what happens is that the NAM pentapeptide is grafted onto the bactoprenol, but to give you a diphosphorylated bond here. So what is the other product of this reaction? What happens to this? What does this give you? Because I didn't put it on the diagram, but you know, if you think about it, this has got to leave, and it will be what? Uridine diphosphate. Everybody got an idea of what that is? Everybody can visualize ATP. Yeah? ADP, everybody sees that? Okay. Well, this is the same except with uridine. Okay, so it's like got a nucleotide base and then 2 phosphates. Once the NAM has been grafted onto the bactoprenol, what have I got? What does this become? Yeah, so this is giving up 1 phosphate here, right? But then what happens to this? Yes. Okay, so this, if you could, like, if I had an arrow here, would be UMP. So one of these phosphate groups here is being given up to form this bond. And that's interesting because that means you've got a little bit of energy here in this bond. And it actually came from here to begin with. Okay, so this structure is also called lipid 1 in the biosynthesis pathway of peptidoglycan. So the next step is what? The next step is adding on the NAG, NAG. And this is called lipid 2. So this is our repeated structure. So we've got lipid 2. This is the lipid part, NAG, NAM, pentapeptide. And what happens is that this whole thing is going to be flipped in the membrane by an enzyme. So the gene is called MUR-J. And that's going to flip the whole thing over so that you've got the NAM-NAG pentapeptide in the periplasm now. Then what happens is that transglycosidases can add this NAG-NAM to the end of another chain, of another polymer. And you've got 3 ends here because you have the autolysins that are cutting the peptidoglycan up all the time. So you're going to add this NAG-NAM to the end of the polymer. Now, here, you're forming a new glycosidic linkage, right? So normally that takes energy. But at the same time, you're forming a bond between this NAG and this NAM. You're breaking a bond between the NAM and the phosphate. So it's neutral. Okay? So this kind of phosphate linkage means that you've like exported the energy that's required to make the glycosidic bond. Now what happens next is that, oh, come back. The diphosphorylated bactoprenol is flipped over again in the plasma membrane, concurrent with it losing a phosphate, and this regenerates bactoprenol monophosphate facing into the cytoplasm. So it's available for another round of synthesis. And the last step is the transpeptidase reaction. So here you've got a free amine group on the side chain of lysine or the diaminopimelic acid. And it's going to form a peptide bond with the fourth D-alanine here. So what's going to happen is that you'll get a peptide bond formed between this carbon and this nitrogen. And at the same time, you break this peptide bond and liberate one D-alanine. So once again, it's energetically neutral. You form one peptide bond and you break one peptide bond. So it can work, it can actually run. So that's the whole reason why bacteria synthesize the precursor as NAG-NAM pentapeptide, because the very last peptide bond is the thing that's carrying the energy that allows the transpeptidase reaction to work in the periplasm. So at the end, this D-alanine will be re-imported back into the cytosol and reused to make more peptidoglycan. Okay, what time is it now? Oh, we're doing okay. So I'll just maybe go through these last two slides to summarize the differences in gram-positive and gram-negative cell walls. So this is a diagram of a standard gram-positive bacterial envelope, and the envelope means peptidoglycan plus plasma membrane. So you've got the very thick sheet of peptidoglycan, and the only other component that you find in all gram-positives is teichoic acid and lipoteichoic acid. So teichoic acid is a polymer of glycerol or ribitol phosphate, and R is some kind of functional group. Could be a sugar, could be an amino acid, different depending on the species of bacterium. But in any case, you've got a long linear polymer of phosphoglycerol or phosphoribitol. And of course, phosphate, you've got a lot of negative charge. That's why it's acidic. So these polymers of lipoteichoic acid are covalently attached at one end to peptidoglycan, and they form a kind of long tube that is making electrostatic interactions with different layers of the peptidoglycan. So it's kind of holding the whole thing together so that you don't have layers of peptidoglycan at the top detaching from the bottom. It's kind of strapped it together with the teichoic acid. Now, lipoteichoic acid is the same thing, except at one end it's attached to a lipid which is inserted into the plasma membrane. And that's important because that's going to bind this whole peptidoglycan layer to the plasma membrane so it doesn't just like shear off. So that's basically what you find in gram-positive cell walls. In gram-negative cell walls, the main thing is you've got a thin layer of peptidoglycan and an outer membrane. Now the outer membrane is interesting because the outer leaflet isn't composed of a regular phospholipid, it's composed of lipopolysaccharide, LPS. So lipopolysaccharide, where you've got one lipid part here, which forms the membrane, and the polysaccharide forms, I suppose, what you could call a kind of layer of polysaccharide all the way around the bacterial outer membrane. Yeah, essential, all gram-negative bacteria have LPS. Now, a lipid bilayer is impermeable to hydrophilic small molecules like glucose, amino acids, and bacteria will probably want to get some of these and molecules inside the cell. Also impermeable to salt, chloride, ions, and sodium, potassium ions. So in order to have some permeability, all gram-negative bacteria have to have porins in the outer membrane. Otherwise, they won't be able to import or export anything. So porins, all gram-negatives have them. And the other protein that you will find in all gram-negative bacteria is Brown's lipoprotein. And here it's got the protein part interacts with the peptidoglycan, and the lipid part is inserted into the outer membrane. So once again, this is a structural component. Its function is to stop the outer membrane from detaching from the peptidoglycan. It's just like an anchor holding these two things together. Okay, so I think that's probably a good place for me to stop right now. So we can come back in 10 minutes. I'll go get a glass of water and we'll try and start up again at 3:25.